Hey guys, so today I wanted to give a quick lecture about how the inner ear works, right? How the cochlea is able to transduce or convert sound into action potentials, and then how the vestibule is able to allow us to feel our position in space, and how those semicircular canals are able to allow us to feel rotational acceleration. So, what I'm going to do here, let me shrink this down, put this right over here. So here's a little drawing that we produced in class. This is an overview of what the ear looks like. You've got that external ear, which includes the auricle and the ear canal. That ear canal is about 2 to 2.5 centimeters in length. Then at the base of the ear canal, that's where you have the tympanic membrane. Okay, it's the eardrum. On the other side of the tympanic membrane, that's when we have the ossicles. Those are labeled the malleus, the incus, and the stapes. Their job is to convert vibrations of the tympanic membrane, which was produced by the sound entering the ear canal, convert those vibrations of the tympanic membrane into vibrations in bone. They also magnify or amplify those vibrations or that signal. The last ossicle, the stapes, will connect to the entrance to the cochlea. That's called the oval window. That's what OW stands for. And then the job of the cochlea, it's a fluid-filled structure that converts these vibrations into action potentials that are sent to the brain through this cochlear nerve right here. That's what all of those green lines represent. Okay, here you have the vestibule. The vestibule is kind of like the head of this snail-shaped structure. Its job is to convert, or is to allow us to feel our static position of our head in space, whether our head is upright or facing downwards or up. It also allows us to feel linear acceleration, such as when you're in a car and it accelerates, or if it puts on the brakes. It also allows us to feel acceleration in a vertical direction if you're in like an elevator. These semicircular canals right here, those are going to be responsible for allowing us to feel rotational or spinning movements, rotational acceleration or spinning movements. One thing that I did want to mention is that I drew these parts of the middle and the inner ear way too large compared to the auricle. I did that just so you could see it a little better. To put that into perspective, if we were to kind of make them the right size, they would be about this size. Let me kind of pull this over. So that's like the actual kind of size of the middle and the inner ear relative to the ear canal, but I just kind of magnified them so that we could see those structures a little bit better. All right, so let's get that out of here so we can get back to talking about what we need to talk about. All right, so one thing that's important is that the specific cells that allow us to feel or encode sound in the cochlea and give us our sense of balance, they're called hair cells, and they're these really special cells, and they look like this. So here is a drawing of a hair cell. Let's focus on this drawing on the left right here. The hair cells are going to have a cell body, okay, and then extending with a nucleus, that's what that circle represents, and then extending upwards from the cell body, you're going to have these hair-like structures. That's what gives it its name, hair cell. This purple, longer extension is called a kinocilium, and then these pink, shorter projections, those are called stereocilia. And at the very ends of these cilia, you have these little red lines, like here, 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 and here. Those are called tip linkages, and what they are is they act like little doors that will allow ions like sodium to enter into the cell body of the hair cell when those tip linkages are opened. Okay, so at rest, this is a hair cell that is not being deflected. Obviously, things will kind of bend these cilia back and forth. This is a hair cell at rest, and these cilia are not being bent. So at rest, there's a tiny opening between the tip linkages, like right here. And that's going to allow a little bit of sodium to enter into the cell. That's going to cause a slight depolarization, causing this hair cell to release a little bit of neurotransmitter, in this case glutamate. This little bit of neurotransmitter will stimulate the connected afferent neuron here to fire a couple action potentials. So at rest, this hair cell is firing a couple action potentials by itself, or causing a couple of action potentials in the connected afferent neuron. If the cilia are deflected to the right in this direction, that opens up the gaps between the cilia. And that will allow much more sodium to enter the cell, causing a much more dramatic depolarization, which will then trigger the connected afferent neuron to fire a bunch of action potentials back to back to back. If the hair cell is deflected in the other direction, you'll notice that the openings between the cilia are closed. They're not open. Those tip linkages are completely blocking those little passages. No sodium enters into the cell, and that causes 0 neurotransmitters to be released. In turn, the connected afferent neuron does not encode an action potential. So just to summarize here, hair cells are stimulated when they are deflected, and they can differentiate which direction they are deflected. If they're deflected in one direction, it causes a dramatic increase in action potentials in the connected afferent neuron. In the other direction, it causes a dramatic decrease or no action potentials to be produced in the connected afferent neuron. All right, and these are the cells that are responsible for allowing us to hear and feel balance. Let's see exactly how this works. So let's move this out of the way here, and let's go over to the vestibule. Let's talk about that first. So in the vestibule, there's actually 2 different structures. The first structure is called a utricle, or the utricle, and the second structure is the saccule. In the utricle, if we zoom in on that, the utricle consists of a bunch of little hair cells that are arranged in a horizontal kind of array. That's what all these little black lines and circles represent, or hair cells with the cilia projecting upwards. The tips of the cilia insert into this gelatinous matrix that contains tiny rock-like structures called otoliths. And what happens is if you experience linear acceleration, like if you're in a car and the car accelerates really quickly, the hair cells accelerate with your head because they're connected to your head, but there's going to be a delay in the motion of this connected otolith due to inertia. That differential motion between the hair cells and the otolith above it causes a deflection of the hair cell, like as it moves back and forth, kind of like that, and that tells your brain that you're accelerating. If they deflect in the other direction, that's deceleration. Now, the saccule is nearly identical in structure, it's just arranged vertically, and this allows you to feel acceleration in the vertical axis. So if you're in an elevator and it drops suddenly, the hair cells will drop, the saccule is kind of kept in its position, and that will stimulate action potential sent to the brain. Okay, so that's kind of how that works. Now let's talk about the cochlea. So the cochlea is the structure that allows us to convert sounds into action potentials. It's this coiled up tube, fluid filled. If we were to unravel the tube, you'd notice that it's going to look kind of like this. So this is like the cochlea being unraveled. Also, if you were to cut the cochlea in half and look inside, you notice that there's going to be 3 little chambers in the cochlea. The chamber on the top is called the vestibular duct, the chamber in the middle is called the cochlear duct, and the chamber in the bottoms is the tympanic duct. At the base of the cochlear duct, that's where you're going to have a series of little hair cells. They're actually covered underneath a special membrane called the tectorial membrane. I haven't really drawn that here, but in the cochlear duct or in the base of it, you have these little hair cells. And if we look at this other view of the cochlea kind of stretched out, these hair cells are going to extend from the base of the cochlea all the way to the very end. That stapes, which is the last ossicle, is going to connect to the beginning of a vestibular duct. And as it beats back and forth when it's vibrated by sound, it's going to convert that, or it's going to transmit those vibrations to vibrations in the fluid that's going to travel through the vestibular duct. Now, at some point along the cochlea, those vibrations of the vestibular duct are going to be perfectly tuned to resonate with some part of the cochlea. High-pitched sounds are going to resonate with a part of the cochlear duct near the entrance of the cochlea, whereas low-pitched sounds are going to resonate with a part of the cochlear duct that's near the end. And when it resonates with that particular portion of the cochlea, it's going to cause the vibrations to transmit through the cochlear duct, and it's going to cause those hair cells to vibrate back and forth. Well, we know what that's going to do. It's going to stimulate those hair cells to send action potentials to the connected afferent neurons. When they reach the brain goes, oh, well, I received those action potentials from neurons that connect to the hair cells in the very end of the cochlea. That means it's a low-pitched sound. If it's a lot of action potentials, it's a loud sound. If it's a few action potentials, it's a soft sound. So that's basically how the brain can differentiate between high-pitched and low-pitched sounds. Now, that's kind of how the cochlea works. Let's go over to the semicircular canals. Each of these semicircular canals are going to be filled with fluid. And as you spin or rotate in different axes, that fluid is going to spin throughout these semicircular canals. At the base of the semicircular canals, you're going to have these special structures. Each one is called a crista ampullaris, and these exist, or they're composed of a bunch of different hair cells that are arranged almost like, it looks like a bud of a flower or something like that. So if you start spinning, fluid in one of these semicircular canals is going to start rotating. As it rotates, it moves past the crista ampullaris. It deflects that crista ampullaris, which is going to stimulate those hair cells, sending action potentials in the connected afferent neurons. Those connected afferent neurons are going to make up the vestibular nerve, and that is going to combine with the cochlear nerve to create the vestibulocochlear nerve. Right? So that's kind of how it all works. It's all based on hair cells. One tricky thing about these hair cells is they are fragile, and loud sounds, especially in the cochlea, can cause these hair cells to die. And in humans, once we lose a hair cell, they don't grow back. In fact, if you, after hearing something really loud, if you hear ringing in your ears, that's because the hair cells have died and the connected afferent neurons don't really know what to do, so they just fire spontaneously, and that's tinnitus, right? So hopefully this helped clarify a couple of things. Thank you.